Good morning. In this second video, we are going to discuss the capacity of the nucleus, its important role in providing the genetic information for producing substances or synthesizing substances inside the nucleus, inside the cell. So we know that active cells should be small. And why should active cells be small? Because the smaller the cell, the larger the surface area to volume ratio. So active cells are usually smaller in size. They have smaller radii in order to allow a larger surface area to volume ratio. So whenever we have an active cell, this cell should be small. Metabolically active cells like liver cells, for example, are small cells. And sometimes metabolically active cells like the muscles, for example, are large. They have to be large because a muscle fiber has to be elongated in order to accommodate or to be proportionate with the distance between the origin of the muscle and the insertion of the muscle. However, muscle cells are active, and in order to work out this issue of a cell being active and at the same time forced anatomically to be large, then this issue will be worked out by providing the cell with multiple nuclei. So metabolically active cells are usually small, but if the cell is forced to be anatomically long, just like the muscle cells are, then there should be a strategy in order to work out this issue, and it will be a multinucleated cell. And that's why muscle cells, skeletal muscle cells in particular, because they are elongated, they are multinucleated. The nucleus, as we said before, contains, in this first paragraph, contains the genetic information, the DNA molecules that are divided into smaller sequences called the genes, and those genes code for particular proteins, and those proteins will be the enzymes that catalyze chemical reactions, the hormones that regulate chemical reactions, the antibodies that help in protecting the body, and so on. And that's why we say that the nucleus should be able to provide enough genetic information for adequate quantities of substances, adequate quantities of enzymes, hormones, and antibodies. So the nucleus is responsible for controlling the synthesis of all important molecules in the cell. And it should be up to the standard. It should be able to cope with the requirements of the cell, either by being very small and therefore increasing the surface area to volume ratio, because the surface area is the factor that will limit intake of substances required for the chemical reactions and output of products produced by the cell. Sometimes there are certain organisms like the protozoan Paramecium. We know that protozoa is the general name for one-celled or unicellular but eukaryotic organisms, meaning they have true nuclei. This is different from prokaryotes, which are unicellular but prokaryotic, of course. The Paramecium, for example, it's a freshwater and ciliated organism, and it possesses two nuclei, a large nucleus, as you know, and a smaller nucleus. The Paramecium has two nuclei, a macro nucleus and a micronucleus. This is another example of cells that might contain more than one nucleus for more control of more metabolic activities inside the cell. There is a fungus called the slime mold, and that consists of one giant cell containing thousands of nuclei. So we can see the outer layer of the protoplasm, which is the ectoplasm, and the inner layer, which is the endoplasm with multiple nuclei inside. This indicates you that this cell is metabolically active. For example, in case of the paramecium, it's understandable that the cell is metabolically, the organism actually is metabolically active because it has cilia, and those cilia have to move, and the motor proteins utilize energy and ATP for movement of the organism in freshwater. And now we come to an overall view of the phases of the cell cycle. As we said before, we have the interphase, which is the longest phase, and that starts here in the cycle and goes until we start the mitosis phase. So interphase, as we said before, consists of the G1 phase, the S phase, and the G2 phase. And as we said before, G1 phase is growth 1 phase. And in such a case, there is growth of all the contents of the cell. The cytoplasm is growing up, the organelles are growing up, and the cell is absorbing, of course, a lot of inorganic and organic materials in order to be able to achieve this growth. But the only exception is the chromosomes, meaning that the chromosomes will not increase in amount. The DNA will not increase in amount, will not grow up. Of course, the DNA molecules do not grow up in length, meaning that if a DNA chromosome consists, for example, of 30 subdivisions, which are the genes plus a bit of non-coding DNA, it will stay as it is. But when we say growth of the chromosomes, we mean what occurs in synthesis, which is the duplication of the chromosomes, meaning that each one of the 46 molecules of DNA, which are not yet considered chromosomes, will be duplicated, and we will have 92 molecules. So if we graph time and content of DNA, so for example, in growth 1 phase, if this is the content, this bar column DNA molecules, then in the S phase, we will have double this amount, which is 92. So as time passes from growth 1 to synthesis phase, the chromosomes are duplicated. So this is S is the phase where the chromosomes, or actually the DNA molecules, are duplicated and they start their condensation process. And then in the G phase, in the G2 phase, there will also be growth, further growth of the cells, but there will also be a double check from the part of the cell for the duplicated chromosomes in particular. We are very concerned with the duplicated chromosomes because they carry the code or the genetic material that will determine the accurate and correct synthesis of all the proteins in your body. And then when everything is okay, when the check mechanisms in the cell say that everything is okay, that there are no errors, or that all minor errors have been repaired, because we might also repair some repairable inadequacies of DNA. So we start the phase of mitosis, and mitosis itself consists of those 4 phases. Prophase, you know that pro means before or the beginning, and then metaphase, which comes after, and then anaphase or anaphase, and telophase. And then after all these phases of mitotic division, mitosis actually represents the changes that occur to the chromosomes themselves and the division that occurs in the nuclear material. We start cytokinesis, which is the division of the cell into daughter cells. So mitosis is actually the division that occurs in the chromosomes or in the nucleus, the changes that occur to the chromosomes in the nucleus. So it is actually nuclear division. Mitosis is nuclear division, but cytokinesis is cell division, the final cell division into two daughter cells. So let's recapitulate the phases of the cell cycle. We have G1, or a period of intense growth and biochemical activity. And of course, this intense growth requires the uptake of material from the surrounding environment, and at the same time, it requires getting rid of waste products of metabolism. So it's a period of intense biochemical activity. And then S, where we have synthesis of new sister molecules of DNA. So it is actually the phase of replication of DNA. And then we have G2, where the cell continues to grow. And at the same time, there is a double check for the status of the DNA molecules and the chromosomes. So here we double check before going into mitosis. Now, let's see how the cells look like under the microscope, under the high power of the light microscope. In interphase, this is a section from the root tip of an onion plant. Why from the root tip? Because at the tip of the root, just like at the tip of the stem, we have a very high rate of cell division, of nuclear division or mitosis, and cell division or cytokinesis. So you can see here that some cells are already in the interphase, but other cells are in different phase. So not all the cells are in the same phase. For example, in this cell, you can see that we can't see a distinct nuclear membrane, for example. So let's see how the cell looks like in interphase. You have a clear, well-defined nuclear envelope. You have a clear, visible, shiny body under the microscope, of course, called the nucleolus, where there is assembly, as you know, of ribosomal RNA with the nuclear proteins that make the ribosome. And of course, we still don't see visible chromosomes. We just see visible chromatin or DNA and nuclear material. Usually the nucleus is this is too big to be the nucleolus. I think this is the nucleus, and the nucleolus is a very small body inside. Yes, this is the nuclear envelope, I'm sorry, and this is actually the nucleolus. Compared, of course, to the size of the nucleus, it makes sense. Now, this is the nuclear material, and under the microscope, actually, the nuclear material is basic, meaning that you see it more or less blue. So this is the phase of the interphase, because you see a distinct nucleus, a distinct nuclear envelope, a distinct nucleolus, but you don't yet see chromosomes. The interphase is the longest phase of mitosis, as we said before, and 90% of the time of the cell cycle is spent in interphase. So this is important to know. You're frequently asked about this. And then it is the phase where DNA doubles, or DNA molecules make accurate copies of themselves. When we say DNA replicates here in interphase, we don't yet have chromosomes, but we can say that the DNA molecules make accurate replicates or accurate copies of themselves. Meanwhile, protein synthesis also doubles, of course, because when DNA doubles, protein synthesis will double. Of course, the organelles will duplicate, and all this is because the cell is preparing itself that at the end of mitosis or at the end of the cell cycle, the daughter cell, we will have two daughter cells from the mother cell. So we should have enough genetic material for the two daughter cells and enough proteins and other components for the organelles and the cytosol. But as I have just said, we don't yet see distinct chromosomes. The DNA appears as uncoiled. And when we say uncoiled here, we mean non-condensed chromatin threads. And we said before that chromatin is a mixture of DNA plus the nuclear proteins, which are the histones and the non-histones. And why do we say threads? Because they are still expanded, not folded, not highly folded, not condensed, and they are still very thin, of course, because they get thicker when they condense. They become visible only when they condense, because when they condense, they become shorter and they become thicker. So until the process of condensation occurs, they are threads, they are thin, they are elongated, they are expanded, they are not folded, they are not yet short and thick. And we can see one or more nucleoli in the nucleus, of course, according to the activity of the nucleus. The centrosome, if you remember, is present at the pole, one of the poles of the cell, as two cylindrical centrioles that are perpendicular to each other, and the centrosome starts duplication. When does the centrosome start duplication? During the S phase. And by the way, it applies to all the structures that have been mentioned to have duplicated, like DNA and like the molecules protein duplicates and the organelles duplicate and the centrosomes duplicate. All this appears during the duplication phase or the S phase. And at the end of interphase, the centrosomes start their migration toward the duplicated centrosomes, start their migration towards the poles of the cell during the G2M transition. What is the G2M transition? It is the transition between the second growth phase and the beginning of mitosis. Mitosis consists of the actual division of the nucleus, not of the whole cell, and sometimes division of the duplicated chromosomes, especially in mitosis. The duplicated chromosomes will separate during anaphase. So it's actually a separation of the chromosomes, a division of the nucleus, and we call it karyokinesis. So we have karyokinesis, where karyo refers to the nucleus, and then following it, we have the final cytokinesis, or division of the cytoplasm into two cells. Mitosis is a continuous process, meaning that the phases are continuous with each other. But scientists, for making it easier, for making our life easier, divided them into 4 phases. But there are no clear distinctions between the 4 phases. They merge into each other. Prophase is divided into early prophase and late prophase. And now we move on to early prophase. Let's first see what the stages of early prophase are. What are the events that occur in early prophase on the diagram and in the text before seeing the microscopic appearance and how we see the cells under the microscope. In early prophase, chromatin, which is DNA plus nuclear proteins, becomes condensation, meaning that it becomes shorter and thicker, and therefore it becomes visible. So we move on from the stage of chromatin, extended chromatin, to the stage of condensed visible chromosomes. And when they become visible, we can visualize them and we can see that every chromosome is actually a duplicated chromosome, as we said in part 1 of the video, and every one of the duplicated chromosomes consists of 2 sister chromatids. Every one of the duplicated chromosomes consists of 2 sister chromatids, and the 2 sister chromatids are connected together at the centromere. And take care not to be confused or not to mix up the word centromere and the word centrosomes. But we can still see the nucleolus. In the stage of early prophase, we can see the nucleolus. Maybe it's not visible here on the diagram, but under the microscope, it should still be visible. Actually, we don't see here the nucleolus, meaning that this diagram might be a bit later than early prophase, but the nuclear envelope becomes fragmented, meaning that it starts degeneration, and it's the phase before complete disappearance of the nuclear membrane. You can see, of course, that in early prophase, the centrosome is already duplicated, and that each centrosome consists of 2 centrioles, and that they begin their migration towards the poles of the cell. So if we go back to the microscopic appearance, we can see that this is early prophase. Here it is clear that the nucleus is not as visible, or the nuclear envelope, the nuclear envelope is not as distinct and discrete and visible as the interphase, and this is for one simple reason, that the nuclear envelope begins its fragmentation process. And here, for example, we can see a visible nucleolus. So the nucleolus is still present. And as you can see, the nuclear material or chromatin becomes more and more condensed, but it's not yet, in this diagram I've just shown to you, it's not yet visible or clear that we can see visible chromosomes. So in this diagram of early prophase, we don't see visible chromosomes, but we see condensed chromatin as an introduction to the visibility of visible chromosomes. Again, we are still in early prophase in animal cells. We are focusing here in this diagram on the mitotic spindle. And the mitotic spindle arises from the centrosomes, as you know, because every centriole, as you know, consists of microtubules, and those microtubules will proliferate and nucleate in order to become longer and longer. And since we have microtubules that originate from every centriole, from each of the 2 centrosomes and the component centrioles, then they will meet each other. As they emerge from the centrioles, they radiate a little bit, they diverge a little bit at the beginning, and we call this the asters. So the asters are the structures of the mitotic spindle that initially appear from the centrioles. And then they join together, and actually, before joining together, the chromosomes get attached to them. So we have asters, and then the asters develop into a mitotic spindle, and it is here that the chromosomes will be attached, as you know, at their kinetochores, and this is very important for accurate cell division, for preventing chromosomal mutations. Okay, so as we said before, the centrioles start their duplication at the interphase and then start moving at the poles of the cells. And as they move at the poles of the cell, at opposite poles of the cell, the microtubules lengthen and lengthen, meaning that, for example, if I have one centriole, one of the two centrioles in this pair and another one in this pair, and they are joined by their microtubules as they go farther from each other towards the opposite poles of the cell. These microtubules nucleate more and more and lengthen. And now we have the stage of late prophase. And in late prophase, as you can see, the nuclear envelope has completely disappeared. The nucleolus has completely disappeared. Here, we can still see remnants of or remains of the nucleolus. Maybe we can see parts of the nuclear envelope, but in late prophase, all these structures that started degeneration in early prophase have disappeared, and we are moving now to the late prophase, in which we have visible, distinct chromosomes, and they are duplicated chromosomes, no nucleolus, no nuclear membrane. So in late prophase, the nuclear envelope breaks up, the nucleolus disappears, and the microtubules, which started, by the way, as asters, and then they nucleated, and by nucleated, I mean that the molecules duplicated themselves and they became longer polar fibers, as we explained on the previous slides, as the centrioles move away from each other. And these polar fibers will reach from each cell pole to the equator, meaning that if I have one centrosome here and another centrosome there, and they are present at the two opposite poles of the cell, which becomes a bit more or less oval or elongated, and these polar fibers are microtubules that extend from the centrioles, as we can see, and they lengthen until they reach the cell equator. And here, in the cell equator, this is the position where the chromosomes will be aligned in metaphase, because in metaphase, this is the stage where all the chromosomes will align. When we come to the structure, or when we review the structure of the chromosome, we have here the centromere of the duplicated chromosome, but we have here the outer kinetochore and the inner kinetochore on both sides. So the fibers, the polar fibers, which will join the kinetochore, actually, will be called the polar spindle fibers. The polar spindle fibers, I'm sorry, that join to the kinetochore are called the kinetochore spindle fibers. The importance, so the kinetochore spindle fibers are part of the spindle fibers. Not all the spindle fibers are called kinetochore spindle fibers. Only those ones that are attached to the kinetochore are called kinetochore fibers, and they are very important to adjust the position. Why is it very, by the way, this is one of the events, that the important events in the cell, alignment of the chromosomes on the equator by the kinetochore spindle fibers, because if this alignment is not correct, we might have an incorrect division of the chromosomes or the sister chromatids on the two daughter cells, and this might result in a chromosomal, not a gene mutation. And we will see very soon in the unit on heredity the difference between a chromosomal mutation and a gene mutation. A chromosomal mutation is just an incorrect number of an incorrect division of the sister chromatids that results in an incorrect number of chromosomes in each of the two daughters. Now we move on to the stage of metaphase. And as you can see from the slide, it's very clear that here the chromosomes are aligned at the equator of the cell. And this is a very important step before the next stage, which is anaphase, when the sister chromatids, as you can see, separate towards the two poles of the cell. So in metaphase, the centrosomes are already at the opposite poles of the cell. There are spindle fibers that arise from the centrosomes and lengthen until they reach the kinetochores in the centromeres. And those spindle fibers that attach to the kinetochores, are called the kinetochore spindle fibers. Of course, by the time we reach metaphase, the nuclear membrane is completely disappeared. Sometimes it disappears even before we reach metaphase. It disappears in the late prophase. And a very important, one of the most important events in mitosis is the lining up of the duplicated chromosomes as a single file along the cell's equator, which we call the metaphase plate. This is the metaphase plate. It is the cell's equator at which the duplicated chromosomes align. Why do we say as a single file? Because in metaphase of mitosis, every duplicated chromosome stands alone. And if, for example, the total chromosomal number, like in human beings, is 46 chromosomes, then you will have a long queue of 46 chromosomes that are aligned at the equator of the cell. So it's a single file, not like or as it occurs in meiosis, where we have a double file, meaning that every two similar chromosomes will align next to each other. But in case of mitosis, metaphase of mitosis, they align as a single file along the equator plate. And then notice that in the stage of metaphase, in order for the spindle fibers that arise from the centrioles to reach the equator plate, they lengthen in order to reach the kinetochore of the chromosomes and adjust the position of the chromosome. But after having done this, after having made sure that these kinetochore spindle fibers have been properly attached to the chromosomes, the next step is not an elongation step. So we have first an elongation step in order to be able to reach the equator of the cell, but in order to achieve the next phase, which is anaphase or anaphase, both are correct, we have to like pull onto the sister chromatids. So actually, at the beginning of anaphase, or what makes the separation of the sister chromatids possible in anaphase, is the shortening of the spindle fibers, because when the spindle fibers shorten, they pull onto the sister chromatids. So the centromere here that used to join the two sister chromatids of the same chromosomes will be forced to split, and the two sister chromatids will be forced to move apart. So this actually, it's clearly the anaphase, and in this anaphase, there has been shortening of the spindle fibers in order to separate the sister chromatids. So this is, every one of these is a sister chromatid or a non-duplicated chromosome or a single DNA molecule. And then in telophase, we have early telophase and late telophase. In telophase, we start reversing the process, but not all the steps will be reversed. We will start reforming the cell membrane and reforming the nucleolus, but most importantly, there should be some kind of separation, because now, as you can see in anaphase, by this separation that occurred between the sister chromatids, we have now two identical sets of single DNA molecules. So we are now ready to have two nuclei, and karyokinesis is almost complete. And we need to separate the two cytoplasms in order to achieve cytokinesis. And it depends on whether this is a plant cell or an animal cell. If it is a plant cell, there will be deposition of cellulose in between the two cells to form a cell plate. But this is not yet obviously seen on the diagram because we are still in the early telophase. So in the early telophase, we are very close to forming two daughter cells by separation of the two cytoplasms, by having two prominent nuclei. The nuclear envelope starts reforming and the nucleolus starts reforming, but they're not yet visible, and the chromosomes might still be visible. The single non-duplicated molecules of DNA are still condensed to a certain extent, but as you can see here, they are not really visible as chromosomes. In late telophase, we will see visible chromosomes. So in late telophase, if this is an animal cell, the separation between the two daughter cells will be completely different. We will have like a waste here or a constriction, as you can see, that is done via actin filaments, as you can know, but as you know, but as you have already studied in the chapter or the unit on the cell, that actin filaments cannot do this alone, but they also need myosin motor proteins to assist them. And this is very similar to what occurred in amoeba when amoeba needed to engulf particles. So we start by having a contractile ring, as you can see, as you know from the previous unit on the cell. And this contractile ring is nothing but the actin filaments and the myosin motor proteins. And the result of this contractile ring is the formation of this cleavage furrow, a furrow that is introductory to cleavage or division of the two cells. And here, as you can see, we have unduplicated chromosomes, but we don't yet have a nuclear envelope and a nucleolus. And then moving on, we start having a reforming. So this is not, if this is late telophase, then what you see here is not a nuclear envelope that is undergoing degeneration, but on the contrary, it is a nuclear envelope that is trying to reform and regenerate. As you can see, the astral fibers return, the spindle fibers become shorter again, and at the end of the day, they will not be visible. So in telophase, the chromosomes will cluster at opposite ends, the nuclear membrane and the nuclei reform. Of course, we are talking here about late telophase. The supercoiled chromosomes, of course, we saw the supercoiled chromosomes when we saw the supercoiled chromosomes, they started to condense in prophase, and then they become mostly condensed in metaphase, and then even when they split in anaphase, they are still supercoiled, but they will unravel, they will uncoil, and they will return to their original pre-division conditions, just like they were during interphase as long thread-like strands of chromatin, and I might also add that they are invisible, they are basophilic, as you know, and we are very ready to do cytokinesis. Cytokinesis, as I said, is different in plants and in animals. In plants, there is a lamella that is formed between the two plant cells, but in case of the animal cells, we have a cleavage furrow. Of course, here we still have metaphase, but of course, this is not late. This is metaphase, yes, of course, because the chromosomes are along the equator here. We have different phases of mitosis together. I'm sorry. So this slide shows you the different phases of mitosis together. We have here metaphase along the equator, okay? And we have here a telophase because the two daughter cells have their chromosomes, their single chromosomes that have been reformed, and I can see a faint nuclear envelope around the nuclear material. Cytokinesis, as we said before, is division of the cytoplasm. It begins during mitosis, and it begins during anaphase. Why do we say that it begins during anaphase? Because during anaphase, the cell, the mother cell or the parent cell becomes very elongated in an attempt to divide the two cells into two daughter cells having a considerable size that is enough for them. If it is an animal cell, then it will be a cleavage furrow, as we said, made up of actin filaments assisted by myosin motor protein. So actually the filaments are actin and the myosin are their friend their friend motor proteins, okay? So they are usually associated with each other, just like dyneins and kinesins are associated with the microtubules. But if it is a plant cell, as you can see here, there will be a plate that is formed during telophase, and this plate is formed from vesicles of Golgi apparatus that adhere to each other along the middle line, and then there is deposition of cellulose along them. So we start by the vesicles, and then we have deposition of cellulose and hemicellulose on top of them, and this is the middle lamella. Of course, we have studied the glycocalyx before, and it is this carbohydrate-rich zone on the cell surface, and that were mostly attached, as we said before, to intrinsic plasma membrane proteins, molecules. This is, of course, a recapitulation of what we have said before, and the glycocalyx can be glycoproteins or glycolipids. There are glycoproteins and proteoglycans. They are a bit different, but you needn't know the difference between them. And these glycoproteins and proteoglycans have been secreted into, if they leave the cell, because as you know, there are some types of glycocalyx molecules that stay attached to the cell membrane, but there are other glycoprotein molecules, as we have said before, that are secreted in the extracellular space. And when they are excreted in the extracellular space, they might act as signaling molecules, but they might be another possibility. This we haven't said maybe before, that another possibility is that after being secreted into the extracellular space, they get absorbed again onto the same cell surface as a marker, for example, or onto another surface, onto another neighboring cell. Okay, and then we have also talked about the role of the glycocalyx in contact inhibition, and when the cells come in contact with each other, as we have said before, a signal is generated by the glycocalyx. The signal that is generated by the glycocalyx is the glycocalyx molecule that will leave the cell and signal another neighboring cell. What do I mean by signaling another cell? I mean that this glycoprotein molecule will be secreted by the cell into the intracellular space and then will be accepted by a receptor on a neighboring cell. In this case, it's not adsorbed on a neighboring cell, but it is accepted by a receptor on a neighboring cell. And this is the basis of contact inhibition or density-dependent inhibition. This is how the cells know that they are numerous or that their density is high. When I am a cell and all my neighboring cells receive plenty of glycocalyx molecules from me, then this means that, and I receive plenty of glycocalyx molecules from neighboring cells, then this just means that we are numerous. When all receptors for the glycocalyx molecules are filled with signaling molecules, then this indicates that the number is big. The last slide we're going to discuss on this in this video is the characteristics of the cancer cells. Why am I starting this slide by this is not the case with cancer cells? I mean that cancer cells do not show density-dependent inhibition. Of course, I know this slide is not covering all the characteristics of the cancer cells, but it is only covering some of the important characteristics of the cancer cells. The rest will be sent to you in an important note Word document. In case of the cancer cells, we have three possibilities. Either that the magnitude of this signal is very weak. I mean that cancer cells send a small number of signaling molecules, or that the glycocalyx on the surface and the receptors are degraded. The third possibility that makes, of course, why are A and B important characteristics of cancer cells? Because if the cancer cells do not know that they are very numerous, then they will not stop dividing. Do you remember we had a question in the MCQ talking about the M factor and how it inhibits the replication or mitosis of the cells? And if this inhibition process does not occur, then the cells will continue mitosis forever. The cancer cells actually are immortal unless we give them a medication or we give the patient a medication that stops them. And then they have a third characteristic, which is lack of anchorage dependence. We have said before that being anchored means being attached to something. And usually that something is the basement membrane. So in order for the cells to divide securely, they must make sure that they are standing on a solid ground, and that solid ground is usually the basement membrane, and that's why most of the epithelia and endothelia in the body are single-celled, or they consist of one layer of cells, because this makes them more confident, more secure, and more comfortable. Rarely do cells divide without anchorage dependence. Even when we talk about cells in fluids, like the RBCs, for example, inside the blood, or white blood cells, they do not divide inside the blood. They actually divide inside the bone marrow from mother cells that replicate. So usually we need a basement membrane, but the cancer cells do not need this. And that's why they can divide everywhere. And that's why we can see metastases and secondaries. We can see that we might have breast cancer, for example, and some of the cancer cells penetrating lymphatics, dividing inside the lymph, dividing inside the blood, and reaching other distant structures like bone or the brain. Of course, this is very sad news when you know that the patient has secondaries, because this really complicates the clinical condition. For example, in vitro, on a Petri dish, this is a solid ground the cells can divide on. So we can do cultures, for example, in vitro on a Petri dish. In vivo, this is usually the basement membrane or an extracellular membrane. So cancer cells do not show anchorage dependence and do not show contact inhibition. A and B tell you that they do not show contact inhibition, and C tells you that they do not show anchorage dependence. They are immortal. They divide uncontrollably, without being necessarily anchored to any membrane, and the rate of division stays high even when they are dense. And this absence, as I said, of anchorage dependence is what allows malignant cells to be transported to other areas of the body, resulting in metastasis and secondaries. By having said this, I would have finished mitosis. Meiosis is present on this presentation, the slides are there, but the video that will be sent to you is part 1 of heredity. You will not be forced to watch the full video, which is 1 hour long, but you will only watch the first 40 minutes. It seems that I've explained meiosis with heredity because it is more associated with heredity. So I will adjust the videos on Google Classroom so that you don't get confused. Thank you, guys. And this is part 2, and you will have to watch part 3 and part 4. But part 3 will be the first part of heredity. I will adjust the order on Google Classroom. Thank you.